Hello everyone, welcome back. So today we're going to be going over exercise 5, which is cellular respiration and photosynthesis. Today I will only be covering cellular respiration. So I noticed you guys haven't discussed this in your lecture yet, so I'm going to go into some detail here. And I also want to tell you guys about quiz 4. I will be grading that manually, so I will be changing your guys' scores. I know sometimes if you put, the singular version versus the plural version, it marks you incorrect. So I'll make sure to go through those manually. I'm sorry I haven't gotten to it yet, but that is on my to-do list. So let me just pull up my notes before I get too far ahead of myself. So I actually recorded I thought I recorded this whole lecture, but I forgot to push record. So I just finished doing this. So now I have to redo it. Okay, so I'm going to go ahead and share my screen. And I hope it's recording this time. So there is a lot of material here. Okay, so like I said, we're going to be discussing cellular respiration and photosynthesis. The chemical ingredients for photosynthesis are carbon dioxide, as well as water. So carbon dioxide passes from the air into the plant through tiny pores, and water is absorbed from the soil by the plant's roots. Inside the leaf cells, the chloroplast, it uses light energy to rearrange the atoms of these ingredients to produce sugar in the form of glucose, as well as byproducts of photosynthesis, such as oxygen gas. So these products are used in cellular respiration. So cellular respiration uses oxygen to convert the energy stored in sugars or glucose to another source of chemical energy, which is referred to as ATP, and this will occur in the mitochondria. So in plants and animals, the production of ATP during cellular respiration occurs in the mitochondria. The waste products of cellular respiration are carbon dioxide and water, which, as you can see, are the ingredients used in photosynthesis. So this continues in this pattern. So today we're only going to be discussing activity 1, where we will learn about cellular respiration by measuring CO2 production by yeast. So all living organisms convert carbohydrates, glucose, into usable forms of energy, ATP. So essentially you start with glucose, oxygen, a lot of steps occur. You end up with carbon dioxide, water, a lot of ATP and heat. These many steps or chemical reactions that make up cellular respiration can be grouped into 3 main stages. We have glycolysis, the citric acid cycle, and oxidative phosphorylation. So here is an overview of cellular respiration. You have glycolysis, citric acid cycle, and oxidative phosphorylation. So in glycolysis, you start with glucose and you end with pyruvate. You also generate NADH and 2 ATPs, 2 net ATPs, I should say. And this occurs in the cytosol. Pyruvate cannot enter the citric acid cycle as it is. It has to undergo pyruvate oxidation into acetyl-CoA, and that way it can enter the citric acid cycle. The citric acid cycle generates NADH and FADH2 as well as 2 ATPs. And this occurs in the mitochondria, in the matrix of the mitochondria. And all these high energy electrons, these NADHs, FADH2, NADH from here, get cashed in at this final step. So oxidative phosphorylation. So they get cashed in at the electron transport chain to generate a ton of ATP. So usually this reaction results in anywhere from 36 to 38 ATPs. So we're going to start with glycolysis. So this is the figure you guys are given in your books of how glycolysis occurs. And this is typically how you learn about glycolysis. So step phase 1 is what we refer to as the energy investments phase, and stage 2 is what we refer to as the energy harvest phase. So essentially you start with that 6 carbon sugar glucose. 2 ATPs are required, so ATP required is step 1 and step 3 to form these two 3 carbon sugars. So the 3 carbon sugar formed here is identical to the 3 carbon sugar formed here. So whatever happens here is going to also occur down here. So NAD will be used in the form of NAD and generated, and NADH will be generated. You will have 4 ATPs produced, but remember we need to use 2 ATPs to get this reaction going. So you really only have a net gain of 2 ATPs. Like I said, 1 NADH from here and 1 NADH from here, and 2 pyruvates, 1 from here and 1 from here. And that's really how your book talks about it, but I'm going to break it down in a lot of detail. You don't need to know these structures, these enzymes. I just think it really helps to know all the details to really appreciate what is occurring, and it makes it easier to understand. It's hard to say like, oh, this is what happens in cellular respiration when they only give you like 2 of the reactions that are occurring. So I also think it's important because this is where students begin to really see how their body is working and doing what it does. So this is a great start to really appreciate the reactions that occur in the cell. So 10 reactions are going to occur in the cell to break down glucose. These 10 reactions are very common in all cells, and glucose is the most abundant sugar and the most common intermediate. So like I said, the salmon color is going to be our energy investment phase. This yellow color is going to be our energy harvest or generation phase. So let's go ahead and start off with the first reaction. So this is the first reaction. So here we need ATP to get this reaction going. So you start off with glucose and ATP, and the enzyme responsible for this reaction is hexokinase. So so this reaction is catalyzed by the enzyme hexokinase, and it's essentially going to transfer a phosphate from this ATP to the glucose on position number 6 over here. So one of these phosphates is going to get transferred to position 6 by this enzyme, and this is your end product. So you see this phosphate group here on this 6 carbon position, and this is now reduced to ADP. Our end product is called glucose 6-phosphate. Hexokinase is a flexible enzyme, so it's able to bind to both your substrate glucose and ATP. So think of hexokinase as having its jaws open, so it can bind to both glucose and ATP. It begins to close its jaws, so it's going to bring glucose and ATP in close proximity, so that phosphate can be transferred up here. And then when hexokinase releases them, this is essentially what you get. So this reaction So you need energy to get this reaction to occur because it is not energetically favorable. And you can say like, oh, if you have glucose and just a phosphate and hexokinase, is that enough to get this end product? And the answer is no. You need energy for this reaction to occur. So that cleavage of the phosphate from this ATP releases a lot of energy, and that is the energy that allows this reaction to occur. So a glucose with a phosphate, it's not enough to get this reaction to occur and to end up with this glucose 6-phosphate. You need that phosphate to come from ATP. So glucose 6-phosphate is going to have a lot of energy stored in it because of, this is referred to as chemical potential energy. So in our second reaction, we have isomerization occurs. So if you remember back to week 3, isomerization is basically a rearrangement. So our glucose 6-phosphate is going to be rearranged into fructose 6-phosphate. And if you remember back to week 3, glucose and fructose have the same molecular formula, they just have different structural formulas, and that is what's happening here. This enzyme is basically giving it a different structural formula. So this third reaction is very important because it's a very regulated step. So here we have fructose 6-phosphate. We have another ATP involved, and the enzyme responsible for this is PFK. So this enzyme is going to take a phosphate off of the ATP and put the phosphate on carbon 1 of fructose. So as you can see, a phosphate is taken off of this ATP and put onto this carbon 1 of fructose to make fructose 1,6-biphosphate. So this is our fourth reaction. So here we have fructose 1,6-biphosphate as our substrate. Our enzyme is aldolase. Aldolase is a ligase, so it's basically going to break this into two molecules. So it breaks this into 3-carbon molecules. So we have DHAP and we have a G3P that is formed. So we go from 1 6-carbon molecule to 2 3-carbon molecules. This is very energetically unfavorable as well. So, so how do we make unfavorable reactions occur? Essentially, a cycle can do two things. It can do control concentration, and it can control concentration by two mechanisms. It can reduce increase the reactant, so give you more substrate, and reduce the end products. So reduce these two, and that can make this reaction energetically favorable. Okay, so now moving on to our next reaction. So here, as we saw, we had a DHAP and a G3P that are formed, 2 3-carbon molecules. And now essentially this DHAP is going to go isomerization and become a G3P as well. So here we undergo isomerization and you get another G3P. So this is great for us because now we have 2 identical 3-carbon molecules, and we basically just have to memorize one thing for the rest of the way. So as you can see here, we're here at the step of the reaction. So we just got glucose to split into 2 3-carbon molecules, which is G3P. And whatever happens to this G3P is going to happen to this G3P. So it makes it a lot easier on us in terms of memorization. So now we're going to move on to the energy harvest phase or energy generation phase. So this is where oxidation is going to occur, and this is where ATP is going to be produced. So as you can see, there's oxidation occurring here, and there's ATP being produced here. Oxidation occurs here, ATP produced here, as well as this water molecule. So let's go through that with a little bit more detail. So remember, we have 2 G3P molecules, so this is occurring 2 times in this reaction. So NAD goes in and it becomes NADH. And there is going to be an addition of a phosphate that occurs here. So we are able to add a phosphate. So we're able to add this phosphate in the absence of ATP because of the oxidation that's occurring from this NAD to NADH. This provides us enough energy to put this phosphate on that molecule. So you can see the phosphate gets put on the molecule and NAD becomes NADH. So we're going to cache in all these electron transport molecules in the end to produce a lot of ATP, and this will occur in the electron transport chain. So we're just racking all these NADHs, FADH2s up at this point to really cash them in at the end. So now we're on to step 7. So this is our first production of ATP. So here, so this is going to happen by the transfer of the phosphate group on the 1,3-bisphosphoglycerate to ADP. So we're going to transfer this phosphate group from this substrate to ADP. We're going to be left with 3-phosphoglycerate and ATP. So this reaction makes 2 ATPs because remember we have 2 3-carbon molecules. We have 2 G3Ps. So now we make 2 ATPs and we're basically cutting ourselves even. So we have now broken even because remember we had to use 2 ATPs to get this reaction going. So now we are on step 8. So here, this actually is not a simple isomerization, but for our purposes, all we have to know is that this phosphate group gets moved over here, as you can see here. Hydroxyl group gets moved over here. It's more complicated than that, but we'll just kind of think of it as a isomerization that's occurring, but it's not quite as simple. Okay, so now we have step 9. So our 2-phosphoglycerate is going to become phosphoenol pyruvate, and this intermediate, and this occurs by the enzyme enolase. Water is going to be produced in this reaction, and this this PEP, let me go ahead and pull up my notes real quick because I don't want to misspeak. I believe this has a lot of energy, but I just want to make sure. Okay, yeah, so PEP has so much energy in it. So we're going to really see that in the next reaction. So we start with PEP. And it has so much energy in it that it's actually able to transfer phosphate onto ADP to make ATP. And it has so much energy left in it that it is so close to actually making 2 ATP molecules. It's just a little bit short. So it has so much energy. It can make 1 ATP perfectly. It could almost make a second ATP, but falls a little bit short. So there's still a lot of energy contained in this intermediate. And you will see that in pyruvate. Pyruvate also has a lot of energy left in it, which we will see in the citric acid cycle. So that basically is glycolysis. So we were able to start with glucose and end with 2 pyruvates. And I'm going to go back to this schematic because I just went into a lot of detail. But remember, we start with glucose, 2 ATPs are used in step 1 and step 3. We end up with 2 3-carbon sugars, which is G3P. Here, two NADHs are produced, one from this 3-carbon sugar, one from this 3-carbon sugar. We have 4 ATPs produced, 2 here, 2 here. However, we needed to use 2 to get the process going. So really, we only have a net gain of 2 ATPs and 2 pyruvates are produced. So now we are going to move on to pyruvate oxidation. So in pyruvate oxidation, you start with pyruvate. Pyruvate is going to lose a carbon as CO2, so it becomes this acetic acid. Oxidation of NAD occurs to NADH, and then you have this coenzyme A that comes in, and let me pull up my notes before I forget to say something important. Okay, so yeah, your coenzyme comes in and each acetic acid is attached to this molecule called coenzyme A, which is shown here. Now this coenzyme A can escort this acetic acid to the first reaction of the citric acid cycle. So it's in the form of acetyl-CoA. Once it does that, we will see in the citric acid cycle that the CoA will be released and it'll be able to come back to help this reaction occur. So this is just looking at it a little bit more detail. So we start with pyruvate. We actually start with 2 pyruvates because remember we had two 3 carbon sugars, which led to the production of 2 pyruvates. So we start with 2 pyruvates. Coenzyme A is required and NAD to get this reaction happening. A CO2 is produced and NADH. We also have a lot of cofactors that are used, TPP, FAD, magnesium, and this acid. The enzyme responsible for catalyzing this reaction is pyruvate dehydrogenase complex, and the end product is acetyl-CoA. Acetyl-CoA can now enter the citric acid cycle. So this is what your book shows the citric acid cycle as. So essentially you have a 4-carbon molecule that's already there, and then you have acetic acid, which is your 2-carbon molecule, come in and attach to that 4-carbon molecule. It goes through this cycle. This is what is required. This is your output. 2 carbon dioxides, ATP, 3 NADHs, and FADH2. Remember, there's 2 of these occurring because we have 2 pyruvates that were able to undergo pyruvate oxidation to become acetyl-CoA. So 2 of these cycles are actually occurring. So double of these outputs are produced. And this is the complicated version of citric acid cycle. The citric acid cycle can also be referred to as the Krebs cycle or the TCA cycle. Remember from glycolysis, we got our pyruvate. We had to use NAD and coenzyme A to get this to go to acetyl-CoA. NADH was produced and CO2 in that reaction. Acetyl-CoA can now enter here at what is shown as number 1. So, so it's very important to to point out that these reactions require NAD to occur. And in order to make NAD, you need oxygen. So oxygen is required for all these reactions that I'm talking about that include NAD or FAD. So this pathway is cyclic. There is no beginning and no end. So when we talk about this in class, I'm going to talk about acetyl-CoA being the starting point for this cycle, and that's usually how it's referred to as, but I just want to point out that at any point, this can be a starting point. So this could be a starting point over here. And for example, if you had glutamic acid come in and interact with this enzyme, alpha ketoglutarate, then this would be the starting point. So if glutamic acid was to come in, this would be the starting point for that cycle. So another thing about this cycle is that you are going to have 2 decarboxylations that occur. So you start with a 4-carbon molecule and a 2-carbon molecule to make a 6-carbon molecule, which is called citrate. I'll go over that. But 2 of those 6 carbons will produce CO2 via decarboxylation. So we have a 6-carbon molecule, 1, 2, 3, 4, 5, 6. 2 of these carbons will actually leave in the form of carbon dioxide. So 1 carbon here and 1 carbon here. So then we end up with the 4-carbon molecule. There is no net production of oxaloacetate. So here you start with oxaloacetate and you end with oxaloacetate. You don't have an actual production of this. You start with one, you end with one. 4 oxidations are going to occur. So here we have NAD become NADH. So that's 1 oxidation. Our 2nd oxidation, our 3rd oxidation, and our 4th oxidation. We also have phosphorylation occur, so GDP gets phosphorylated into GTP. And I just want to remind you that all these are generated in doubles. So per glucose molecule. So if you have one glucose to begin all of this, you will have double of these reactions because you have two pyruvates coming in. So just keep that in mind as we go through this cycle. So this is all going to occur in the mitochondria, and that's really why the mitochondria gets the name of the powerhouse of the cell, because oxidation occurs here, so NADH and FADH2. And these oxidation produces a lot of energy. These are really important for ATP production. So that kind of gives you an idea of why the mitochondria is called the powerhouse of the cell. It's because there's so much energy that is produced here. So very briefly, I'll actually talk about this later. I'll bring this up when we talk about the electron transport chain. Okay, so let's go ahead and go through these steps. So I'm going to walk you through all these steps like I did with glycolysis. So here you begin with oxaloacetate and acetyl-CoA, which is shown here. Let me make sure I'm still recording. Okay. And water, and your enzyme is citrate synthase. This so here you're starting with a 4-carbon molecule and a 2-carbon molecule, and it will lead to the production of a 6-carbon molecule. So 1, 2, 3, 4, 5, 6. And the CoA is going to be split out. So you'll see that this CoA is split out, and I kind of briefly mentioned that previously, that the CoA is split out and it's able to go back to that pyruvate oxidation reaction. So this is a very energetically favored reaction. So your cell is super happy to make this reaction occur. And I want you guys to remember that because I'm going to come back to this. So remember that the reaction of oxaloacetate to citrate through citrate synthase is a very favorable reaction that your cell wants to occur. So just keep that in the back of your mind. So the second reaction is going to be citrate to isocitrate. And here, essentially, you're just going to move this hydroxyl group, this OH, to this area. So you're just moving this OH down. This is our third reaction, and this is where our first oxidative decarboxylation occurs. So here we have isocitrate as our substrate, isocitrate as our substrate. Isocitrate dehydrogenase is our enzyme. And when you see dehydrogenase, this essentially means oxidation is going to occur. So here, this is going to require NAD, and NADH will be formed, so oxidation occurs. This is how I learned oxidation. So Leo says GER, lose an electron, oxidation, gain an electron, reduction. So when you lose an electron, oxidation occurs. So we have our NADH. Remember, we're going to cache these electron transport carriers in our electron transport chain for a lot of energy. And this is a oxidative decarboxylation process. So a CO2 will also be generated. So this is just an intermediate, don't really worry about that. So a CO2 will be generated, and our end product is alpha ketoglutarate. So our next reaction is going to be our second oxidative decarboxylation. So essentially the same thing is going to occur. So here the enzyme is called alpha KGDH, and these are the enzymes that are required to make this reaction occur. And if you look closely, these are very similar to the coenzymes that were required for pyruvate dehydrogenase. So for pyruvate to become acetyl-CoA. Okay. So here, the you have the second formation of NAD to NADH. You also have a second CO2 that is formed, and this 5-carbon molecule is going to get reduced to a 4-carbon molecule. And we're going to put that CoA back on. If you can see right here, we're going to make succinyl-CoA. That CoA is going to go back on. And this is a very high energy bond. This bond carries a lot of energy and it's going to come up later on. So that's very important. So our next reaction is succinyl-CoA to succinate. And if you notice this name, succinyl-CoA synthetase is the enzyme that requires this reaction to occur. But this enzyme, the name suggests that in the end you're going to synthesize succinyl-CoA, but actually when this reaction was being studied, it was being studied in the reverse. So it was being studied from succinate to succinyl-CoA. So that's why this enzyme was called succinyl-CoA synthetase, because in that sense it made sense. Succinate was leading to the synthesis of succinyl-CoA, but the name just stuck. So it's kind of confusing when people see this enzyme's name, but just keep in mind that it is going in this direction. So here we have 4-carbon molecule and GDP. So remember we have that very high energy intermediate. So this CoA here had a lot of energy in that bond. It was a very high energy bond. So this very high energy intermediate is going to provide the energy to make GDP into GTP. So it's going to be able to provide that energy to phosphorylate this GDP. So when this bond breaks, the CoA breaks off. When that bond breaks, there's going to be so much energy released that this GDP can become GTP. GTP has the same energy as ATP, so there's a lot of energy here that is being created. So our next reaction, we're going to have succinate and FAD. So here there is going to be oxidation occurring, but this is different from the previous two. As you can see, the oxidation is going to be FAD to FADH2. So all you need to know for our purposes is that oxidation occurs. It's different from the previous two reactions we saw because here it's using a different mechanism. It's using FAD into FADH2. So basically this oxidase is the bond between the two carbons, and we're also going to see that a different electron transport complex, I don't know what to call it, is going to require FADH2 when we cache these in. So just keep that in the back of your mind as well. So here we have fumarate and water. This is a 4-carbon molecule. And this is going to, nothing really important is happening here. It's basically just rearranging this to this hydroxyl carbon hydrogen backbone. It's reorganizing this to this formation, and it's prepping us for our final reaction. It's not a very exciting reaction. And this is our final reaction. So here, the hydroxyl group, the OAce, is going to get oxidized to a ketone, and this requires NAD. So NAD is going to produce NADH. And this is a rare oxidation that is not energetically favorable. So if you remember, how did I tell you that you can make unfavorable reactions occur in a cell? You can do this by regulating concentration. You can either regulate the concentration of the substrate or the concentration of the end product. So here, we're going to take away the end product. So if you remember, when I was talking about this previously, I said that in reaction 1, this substrate gets synthesized into citrate by citrate synthase, and that reaction is very, very, very favorable. So a way to get this reaction to occur is to keep this concentration very low. And the way we can keep this concentration very low is by making it get catalyzed into citrate by citrate synthase and making that reaction favorable. So we encourage the other reaction to keep this low so that we can make this reaction occur because it is energetically unfavorable. So that is now going to make this reaction favorable by doing that process. So let's just go to our quick summary slide. So essentially what you saw is that two carbon dioxides are produced. Remember I said that there was two oxidative decarboxylation reactions that occur. So two carbon dioxides are produced, ATP is produced, 3 NADHs are produced, and FADH2 is produced. And remember, there is double of all this output because we had two pyruvates produced in glycolysis. So two acetyl-CoAs were able to enter the citric acid cycle, so we have double of all this output. So now we're going to move on to the third phase, or the third main step of cellular respiration, which is the electron transport chain and oxidative phosphorylation. So I'm actually going to begin with this part. I told you guys I would talk about this later. So I told you that the citric acid, okay, well first I told you that the glycolysis occurs in the cytosol. Then I told you that the citric acid cycle, the oxidation occurs in the matrix. I told you that all this is occurring here in the mitochondria, and that's why we refer to it as the powerhouse of the cell. And the mitochondria has two membranes. It has this outer membrane, which is porous. It's permeable to oxygen, to gas, to water. Small molecules can pass through quite readily. However, the inner membrane, so right here, it's very impermeable. So most substances cannot pass through, especially things that are polar. So for example, protons cannot pass through. So protons are polar, they're charged. This lipid bilayer is going to prevent the movement of charged molecules coming through. So let me go ahead. So protons cannot really readily pass through this membrane unless two things occur, or there's essentially two ways that it can pass through. And one of those ways is if the membrane's damaged. The other way is through a transport protein, and that's what I'm going to talk about right now. So how can protons pass through? So electrons are stored in electron carriers in the form of NADH and FADH2. Remember I said that we were racking these guys up in order to cache them in here to produce a lot of ATP. So we've been basically just racking these up to cache them in now. And the electrons from both of these complexes, NADH as well as FADH2, can enter the electron transport system and move through this system through these proteins and membranes. I'll tell you guys that. I'll come back to that also. So in the intermembrane of the mitochondria, you have three large complexes. You have complex 1, complex 2, and complex 4. Both of these are essentially showing the same thing. I'm just using this bottom one for reference. But essentially here, this diagram shows complex 1, 3, and 4, but I am going to talk about complex 2 in a little bit. But essentially, your electrons are going to be shuttled through small molecules between these large complexes. So here NADH will donate an electron to complex 1, and complex 1 will donate that electron to coenzyme Q. Coenzyme Q will donate that electron to complex 3. Complex 3 can donate that electron to cytochrome C. Cytochrome C can donate that electron to complex 4. And complex 4 can donate the electrons to oxygen in order to make water. And let's see how I want to explain it. So essentially this is only one, okay. So when NADH donates an electron to complex 1, coenzyme Q can juggle 2 electrons at a time. However, this is the only small molecule that can do that in this reaction. The rest of this can only handle 1 electron at a time. So you really need this to happen at a steady flow in order to not back up this reaction. If you don't have oxygen, you don't have NADH, you don't have FADH2, and this reaction can't occur, and it's going to back it up because the only one that can juggle 2 electrons at a time is coenzyme Q. Everything else can only juggle 1 at a time. So the difference between, so I also told you I'll bring up complex 2. So complex 2 is going to get its electron from FADH2, and it's still going to interact with coenzyme Q the same way. And the only difference is that complex 2 is accepting electrons from FADH. So each time these electrons are moving along, there are protons that are being pumped. And this is very important, the pumping of these protons through these complexes. So they are being pumped from the matrix, where they are found in low concentrations, to the intermembrane space, where they can build up to high concentrations. And that is shown clearly here. So in the matrix, you have low concentrations of the electron transport chain, and they are pumped through these complexes to the intermembrane the intermembrane of the mitochondria. And if you remember, I said that protons can't pass through here. So they need these complexes in order to pass and create this gradient. This gradient is very important in order to allow this molecule to work, which is ATP synthase. So let me see if there's any other information I wanted to say about this. Okay. So this gradient is going to need to occur in order to make ATP. And why is that? Because these protons are going to be able to enter the ATP synthase, and they're going to be what gets this rotating. They're going to get this moving and rotating. And when that happens, ATP can be produced. So I'm going to go through that in a little bit more detail in this image here. So this is our ATP synthase. So now this is a different orientation. So here, this is our matrix. This is the part that is embedded. This is the part that rotates. This is the intermembrane region. And this is that stalk or the mushroom head of the ATP synthase. So the proton will enter through here. This will create movement, and it will create this to rotate. These beta subunits, which are shown here, can have these different configurations. So it can have L, the loose configuration. And in this configuration, ADP and phosphate can bind. In the T configuration, which is this blue one here, ADP and the phosphate are brought in close proximity and ATP is formed. So you can see them being brought in close proximity to form ATP. And in the open configuration, so in the O, ATP is released. So here's the O, this purple one right here. ATP is released. This is the open configuration. So the beta subunits of ATP synthase will rotate between the loose configuration, the T configuration, and the O configuration. And what's making this gamma subunit spin and making in turn this beta subunit spin is essentially those protons that are being pumped in from the intermembrane area to the matrix. So that's why we really wanted to produce that concentration gradient, was to get those protons to be pumped back to get that turning and ATP production going. So this happens really rapidly, and there's actually a release of hundreds to thousands of ATP molecules per second. So there's really nothing quite like this in your body that can create so much energy. This is like a major source of ATP in our bodies, is this electron transport chain. There's really nothing in our body that can produce this much ATP. And I just have this last slide just to kind of tie it all in for you guys. So there are different toxins available that can inhibit this reaction. The cyanide is something that can inhibit this reaction, so the electrons aren't able to be passed on, and it inhibits this oxygen from interacting with the protons and making a water molecule. And when this happens, like I said, when this is being prevented, the electrons are getting backed up. So basically there is no ATP, no ATP production. Well, okay, there's no oxygen, so it backs up the protons. There's no proton gradient now. There's no ATP production. There's no citric acid cycle, and glycolysis stops. So that's why cyanide leads to death. Cyanide poisoning. Also, carbon monoxide is another toxin that can inhibit this reaction. And carbon monoxide really kills you in two ways. It kills you in this reaction, and it also kills you by competing to bind to hemoglobin. So it prevents oxygen from binding to hemoglobin, and so it can really kill you in two ways. But in activity number 1, we're going to measure CO2 production in yeast. So yeast can participate in both aerobic respiration, which is in the presence of oxygen, and anaerobic respiration, which is in the absence of oxygen. So here we're going to measure CO2 by measuring respiration of yeast. So this is our cellular respiration equation. Remember, we start off with glucose and oxygen, and we go through glycolysis, citric acid cycle, the electron transport chain, to end up with 6 carbon dioxide molecules, 6 water molecules, and a lot of ATP. So in this experiment, you're basically going to have a balloon. You're going to have 10 mils of water or sweetener and yeast, and you're going to add that to the balloon. So these are the conditions we're going to have. So we're always going to have yeast. That is going to be a constant. We're always going to have the same amount of yeast, so that's going to be a constant. And we're always going to have 10 mils of a solution. So in our control group, we're going to use 10 mils of water. In our experimental groups, we're going to have 10 mils of a sweetener. So it can either be this artificial sweetener, 10 mils of lactose, or 10 mils of sucrose. So the yeast in the 10 mils of solution will be mixed and added to the balloon. The balloon will be placed in an incubator, which is set at 37 degrees Celsius for 2 hours. And we're just going to let cellular respiration occur. So let's go ahead and propose a scientific hypothesis. So let me give you some background here. So sucrose is made up of what? It's made up of glucose and fructose. So both glucose, so we definitely know that glucose can be used in glycolysis to produce cellular respiration. So that is the input for glycolysis. and glycolysis is step 1 in cellular respiration. So we know that's needed for cellular respiration, and fructose can also be used for cellular respiration. Remember the fructose 6 phosphate and fructose 6 biphosphate, I believe it was, in that in glycolysis. So this will probably produce a good amount of cellular respiration. So we also have 10 mils of lactose. So lactose is composed of galactose and glucose. So yeast they do not contain the enzyme lactase, which is able to break down lactose to galactose and glucose. So yeast are unable to break this down to glucose and galactose. However, lactose can undergo some modifications to be used in cellular respiration. I didn't go through this here, but it can undergo some modifications. However, it requires quite a few modifications in order for it to be used in cellular respiration. So if I had to make a hypothesis, I would believe that cellular respiration would occur in this condition, however, not to the same degree as the sucrose condition. Aspartame is an artificial sweetener, so this is not composed of glucose. So I would not expect to see much cellular respiration occurring in this condition, and likewise, I don't predict a lot of cellular respiration to occur in this condition. This is also going to be our control condition. The water will be our control condition. Okay, so what is the independent variable in the experiment? So the independent variable in this experiment will be our sweeteners. So our independent variables are sweeteners, and the options are sucrose, lactose, aspartame. Our control group is the water. What is a dependent variable? So what are we measuring here? We're going to measure CO2 production. So you could say CO2 production, cellular respiration, any of those would be okay. So how are we going to measure CO2 production? Well, we're going to measure the circumference of the balloon every 30 minutes for 2 hours. So at 0 hours, we're going to take our baseline. We're going to measure the circumference of all the balloons, and they're all 14.5. So we're all starting off the same. At 0.5 hours, we're going to measure the circumference of the balloons. Again, at an hour and a half, and at 2 hours. As you can see, that the sucrose condition led to the greatest CO2 production as we predicted. So which sweetener resulted in the greatest CO2 production in yeast? Sucrose. And which sweetener resulted in the least CO2 production in yeast? Aspartame. Remember, this was an artificial sweetener, and aspartame is shown here in blue. This is an artificial sweetener. It does not have glucose in it. And although lactose cannot be metabolized or catalyzed, cannot be catalyzed into galactose and glucose by yeast, lactose can be used in cellular respiration, just in a different way that I'm not going to discuss here, but it's not going to lead to the same production as glucose and fructose would. So glucose and fructose can really be used in those reactions I talked about in glycolysis to produce cellular respiration, and that is shown here. So this is a picture of these conditions. And what we saw is that the sweetener sucrose resulted in the greatest CO2 production in yeast. So very briefly, I just want to make sure you guys understand these concepts. So make sure you guys know the 3 main phases of cellular respiration, which are going to be shown here. So the 3 main phases are going to be glycolysis, the citric acid cycle, and oxidative phosphorylation via the electron transport chain. You want to know which stages of cellular respiration take place in the mitochondria. So the citric acid cycle and the electron transport chain are going to occur in the mitochondria. Glycolysis will occur in the cytosol. Remember that pyruvate cannot enter the citric acid cycle as it is and needs to undergo oxidation and be converted to acetyl-CoA, where it can enter as a 2-carbon molecule. And if you guys have any questions, please let me know. I know this was a lot of material, so take your time with it. And you really just need to know these general concepts. You don't need to know the detailed concepts. You're just required to know this figure, this figure, this figure, and these figures here. Okay, so I'll put the rest of the PowerPoint up at a later time. So I'll put up photosynthesis and activity 2, 3, and 4 later this week. Please let me know if you guys have any questions, and that'll be it for today.